Hello everyone, welcome to BioWorks. I know it's been long, you guys have been waiting for my next video, but I've been busy, so sorry for the delay. But I thought I'll come up with this topic called dihybrid cross because it seems like it is one of the most confusing and difficult topics in grade 12th and grade 10th as well. So let's hope this video helps you guys. So let's get started. So let's start with today's topic, which is dihybrid cross. So let's first write down the definition and understand the meaning. So it is a cross between two parents differing in two contrasting characters. Alright, so we are going to consider two parents that differ in two contrasting characteristics. And let's say, for example, the two contrasting characteristics are seed color in plants, right? And the other characteristic is seed shape. Fine. Now, when we talk about seed color, there are seeds having yellow color and there are seeds having green color, right? Yellow being the dominant trait over here in seed color, and green is the recessive trait. All right. Likewise, when we talk about seed shape, there are seeds which are round and seeds that are wrinkled. All right. So round seed shape will be the dominant trait and wrinkled seed shape will be the recessive trait. All right. Now we will consider two parents. So let us write down the parental generation here, or the parents. So we will choose one parent which will have seeds that are yellow in color and they are round in shape, crossing with another parent which has green as a seed color and wrinkled seed shape. Alright. So what we have done is simply considered one parent to have completely dominant traits for both the character. Like I said, yellow is the dominant trait for seed color, round is the dominant trait for seed shape. So we have considered both the traits in one parent, and the other parent will have green as a seed color and wrinkled as the seed shape. Both of them are recessive. Fine. Now next step is to write down the genotype. Now what is the meaning of genotype here? Genotype means a set of genes that are present in an organism for that particular trait. Now here we are talking about yellow and round seeds. So the genes that will be present for these characteristics are capital Y, capital Y, capital R, capital R. Capital Y for yellow, capital R for round. We know that genes always work in pairs. So that is why a pair of capital Y and a pair of capital R crossed with small y, small y, small r, small r. So we will consider the for green it is going to be small y, and for wrinkled it is going to be small r, right? These are the genes that determine these traits. That will be the genotype. Next step is to write down the gametes. So let's write down the gametes now. Alright, we generally indicate gametes in circles. So let us write down those gametes. It is going to be YR in one gamete. Similarly, another gamete here, right? And the other type of gametes will be small y, small r, small y, small r. Alright, these will be the gametes form. Now our next step will be to find out the F1 generation. So I'm going to just slide this up a little bit, and let us write down the first filial generation or the F1 generation. Alright. Now to find out the F1 generation, you can either use a Punnett Square or the checkerboard method, or you can use a simply a cross method. So I'm going to show a cross method here for F1 generation, and I'll go for Punnett Square for the F2 generation. Alright, so for a cross method, we simply consider the first gamete here crossed with the first of this one, right? And I'm going to write down as capital Y, capital R, small r. Simple. Now next, I'm going to consider the same gamete with the second one of this one, right? And I'm going to write down the genotype here. Then the second gamete from here with the first one of this, and then the second one with the second one of that one. Alright. Now this is your F1 generation, also called as first filial generation, and what you're going to observe here is that all of the F1 hybrids show same genotype, right? So let's write down the genotype here. Genotype of F1. So all the F1 hybrids show genotype as capital Y, capital R, small r, and the phenotype of F1, we know that phenotype is nothing but the observable characteristic or the physical appearance, right? So the phenotype of F1 is yellow and round. Now why do we say that the phenotype is going to be yellow and round here? Now we see there is a presence of a capital Y and a capital R. Now capital Y and capital R being dominant alleles, they are going to show their own phenotypes. We know that capital Y was for yellow, capital R was for round. So here is the phenotype of all F1 hybrids. They will all be having seeds that are yellow and round. Alright. Now the next step is to carry out selfing of F1. Selfing of F1, which means pollination to be carried out in between the F1 hybrids. Right? So I'm going to choose two F1 hybrids here and write it down. YYRR. This is one F1 hybrid crossed with another F1 hybrid. Alright. Now let me just slide this up a little. Fine. Now here I'm going to write down the gametes for the F1 hybrids. Let us find out the gametes. Now here you will observe that there are four different alleles: capital Y, capital R, small r. There are four different alleles. So hence you will have four different types of gametes that are formed by different possible combinations. So a simpler way to find out the gametes of F1 hybrid is to follow the cross method that we did in the previous section. So here we consider the first Y with the first R and write it down in a gamete. Remember, we write gametes in circle. Then we consider the same Y with the second R. Alright. So we basically combined the capital Y with both the R's here. Alright. And now next we will consider the second Y, the smaller Y, with the first R, and the same smaller Y with the second R. Alright. So this is how we find out the four possible gametes from this particular F1 hybrid. Now we write down the same thing for the second one. Right. Now after writing the gametes, our next step will be to find out the F2 generation, which is the second filial generation. We will find that out based on the gametes. Let us now find out the F2 generation. I'm just gonna scroll this up. Alright. Now we will write down the gametes first in this Punnett square. So let me just label this as F2 generation. Alright, and these gametes that we have found out, we will place them on the table here, right? So let's say first we will write down the gametes here on top, and the other gametes on the side. Alright. Now next thing is to write down the genotype or find out the possible genotypes here. It is done by writing the gametes of the corresponding box. So let's say if I want to write down the genotype in this particular box, I would consider this gamete and this gamete. Alright. So simply I will write down capital Y, capital Y, capital R, capital R. Similarly for here, I will write down capital Y, capital Y, capital R, small r. Fine. So what I did is I simply considered capital Y of this, and capital R and small r. So likewise, let's fill up the entire box here. Alright, so we have written down the genotypes in these boxes. Now next step is to write down the phenotype. So I'm going to write down the phenotype there itself in the box under the genotype. So let's say for the first one is capital Y, capital Y, capital R, capital R. The phenotype is going to be yellow and round. Alright. The second one is also going to be yellow and round. So likewise, I'm going to fill up the entire box by the phenotype. So we have completed the entire F2 generation in the Punnett square, also called as a checkerboard method. Now the next step and the final step is to write down the phenotypic and the genotypic ratio. So to find out the genotypic ratio, or the ratio of the possible genotypes, we will diagonally look at this box. So we will start with this particular box here. Alright. So this genotype capital Y, capital Y, capital R, capital R occurs only once in the entire box. So we will write number 1 here is 2. Now the second one, these two, look at this diagonal, right? This box as well as this box, they have, both of them have capital Y, capital Y, capital R, small r. So it occurs twice. So let us write down number 2. Next, look at this box and this box, this one and this one. Both of them are same, right? They are capital Y, small y, capital R, capital R. So we will write down number 2 again. Next is to consider this entire diagonal. All of them show capital Y, small y, capital R, small r, and they are 4 in number, right? So we write down number 4. Next, we consider this one, small y, capital R occurs only once here. Alright. Next, we consider these two, capital Y, small r, small r. So both of them are same. So number 2. Then we had left out this one, if you remember, right? So because we had considered these two, right? So this box was left. So we write down number 1 for this one. Now we come to these two, small y, capital R, small r. These two are similar, so we write down number 2 for them. And lastly, we are left with small y, small r, which gives us 1. So this becomes a genotypic ratio. 1:2:2:4:1:2:1:2:1. And next is to write down the phenotypic ratio. That is the ratio of the phenotypes. So here we can see there are 4 possible phenotypes. I'm going to just quickly make a note of 4 different phenotypes that we can see here. Yellow and round is one phenotype that we can see. The other one is yellow and wrinkled. Third type we can see is green and round. Alright, and the fourth one is green and wrinkled. Fine. Let's now write down the numbers. So as you can see, if you count total yellow and round phenotypes in the box, you would see it occurring 9 times. 1, 2, 3, 4, 5, 6, 7, 8 and 9. Right? So number 9 here out of 16. So out of 16 possible phenotypes, 9 of them are yellow and round. If you count yellow and wrinkled, you will find them 3 times. So 1, 2 and 3. 3 out of 16 are yellow and wrinkled. If you count green and round, so 1, 2 and 3. 3 out of 16. And if you count green and wrinkle is just once in the entire box, so 1 out of 16. Alright, so that gives us the final phenotypic ratio as 9:3:3:1. This becomes the phenotypic ratio of dihybrid cross. So that was all about dihybrid cross. I hope it was quite clear and simple to you. Thank you.